Bank based over at Hammersmith Hospital in London. Hopefully what I'm going to do in this presentation is give you a quick whistle-stop overview, and I'm here all day today, all day tomorrow, and I'll look to go a little bit more into certain aspects of the brain bank throughout that process too. So, I hope to say who we are, what we do, what our processes are, and sort of the actual level, and a few of the outcomes that we've had from the Parkinson's UK Brain Bank that's been going for about 20 to 25 years now. And please put your hands up, ask questions. There will be images of brains. I do have bits of brains here if anyone would like to have a look later and that sort of thing as well, but I just thought I should give you fair warning. So who are we? Well, we get many names and many different sort of forms, but we are essentially a three-way partnership between the MS Society, Parkinson's UK and Imperial College over in West London. And so what that comes to be is the MS Society Tissue Bank, the Parkinson's UK Brain Bank, the Imperial Brain Bank, or the MS and Parkinson's Tissue Bank. They're all the same thing, we're all the same place, we just have many different names. And we work together essentially to be the largest brain bank in the UK and one of the most internationally recognised brain banks because we have this three-way partnership. Normally there's only one set of funding or there's only one institution involved and they'll fail after a while, but we've been going now for 20 to 25 years depending on how you measure it, and we're one of the most successful brain banks. We're a team both of academics and sort of operational staff. We're led by both a neuropathologist and a neurologist who look after to make sure that we're doing things that are actually in line with what the researchers want to do in the world. And then we use some of the neuropathologists around the country to help report on the brains that we have donated to us. On the operational side of things, there's 6 of us. There's me, and I look after everything outside of the laboratory. There's the tissue bank manager who oversees the laboratory processes, and there's 4 technical staff who look after the entire process for us. They're the clever ones, and they never get let out of the lab, so unfortunately I get all the joy of the South Coast. So a little bit who we are, what we do. This is where we're based at Hammersmith Hospital. It looks like an old hospital. It is an old hospital. It's where the first liver transplant was done. It was where the first sort of MRI was. We don't talk about Aberdeen, who technically got it first, but it's a sort of a hospital that's always been at the forefront, and we continue to do that through our brain banking scheme. So what are we as a brain bank? Well, in truth, we're a donation scheme. And so what that means is when people pass away, they may want to donate tissues to research, and we help fill that role. Just want to differentiate us, and this is normally what I want as a main takeaway, to be honest, when I talk to people about the brain bank is, yes, there's NHS organ donation, and that's the thing you do on your driver's license. You might have a healthy kidney. When you pass away, someone needs a kidney, you can give them a kidney. Fantastic. You also have whole body donation. That's to do with sort of medical schools. That's to make sure that doctors know what they're doing on someone who's not alive before they're let loose on someone who is alive. And that's the donation of the whole body. Both of these schemes, if you sign up to them, you can work sort of in tandem with us, but it doesn't mean you've consented to donate to research. And that's where we fall into this category of research. And so we receive the donation of the brain and the spinal cord, and then we also receive the medical records as well. So we can look at what's happening in life and see how that's reflecting itself in the brain tissue. So we're still trying to understand Parkinson's on that molecular level and try and make those insights, and I'll touch on it a little bit there. But what I also like to point out here is, without the charity, this doesn't exist. If someone with Parkinson's wants to donate their tissue to research after they pass away, can't happen. There's no ethical framework, there's no logistical operation that will allow it to happen. So that's what the Parkinson's UK Brain Bank is born out of. It's this desire for people to donate their tissues to research, and there's researchers needing access to that tissue to be able to make breakthroughs. So that's where it really comes from. It's not supported by these 2 other schemes. What is a tissue bank? This is what a tissue bank is. They recruit people, they might collect some data, and they retrieve tissue. It then ends up on a wall. This is what used to happen in the 1970s, 1980s, and a doctor says, I'll look at that eventually. They have no funding to do it, they don't know when they're going to do it, they've just said, that was an interesting case and we should have a look at it. We're not a tissue bank, we're a research tissue bank. So everything we do, every donation we receive, is to go to research. That's why we have our eligibility criteria, so we have to receive tissue within 48 hours and that sort of thing. And so we do these next steps immediately. We receive that tissue, we perform a diagnosis on that tissue because that's where the buck stops with the pathology, and then we then liaise with the researchers around the world to have access to that tissue. We then get whatever they've done back from them as well. What's important to note, and it's a sort of a nitty-gritty thing, but we're an ethical framework as well. And so if a researcher, Joe Bloggs on the street, says, I want to look at something in particular, in someone who has Parkinson's, look at an area of their brain, they would have to get that approved by an ethics board. They would have to sign up some people. They would have to wait for those individuals to pass away. They would have to have a framework to be able to collect that tissue, and then they would have to do that work. What we do is we look after that process. And so if people say, I want to donate, we receive that donation and then we make it work. And researchers can then apply to us and say, this is what I want to do. And then we have a panel of people living with Parkinson's and scientific peers who say, does it make sense scientifically? And is it in the interest of people living with Parkinson's? If yes, then they can have access to that tissue in an anonymized format. So that's sort of what we do. We're different from clinical trials, and I'll touch on how we're different a little bit later. But I do like to point out that we're more than just tissue banking, this sort of researcher aspect. My most important part is speaking with families of people who have donated, and we provide that feedback to them on what we find in the donation as well. So that means a lot to me, but we're also an educational resource for both doctors and nurses around the country to come visit us and help understand the condition on that sort of actual brain level. And of course, we're a flagship facility for both the charity groups as well. So charity groups come to us and we're something that they've decided to invest in as a resource for research. But where do we fit in the whole research ecosystem? Well, there's a lot of clever people doing things by putting cells in a dish, and they can look at the cells and they can see what's going on. They can give them a little treatment, see how the cells react to it. But the problem with cells is they're not in the human body. So you're missing that interplay. So you've got restrictions there on that sort of in vivo environment. You've also got people that we don't really support using animal models. And animal models are useful because you can give them a treatment and you can see how that might respond to those cells. But the big issue here is, I'm not sure if you guys have figured it out, but a mouse isn't a human. And mice don't get Parkinson's. So they're getting a model of Parkinson's. So everything there it's useful, but it's a hypothetical sort of model. So it doesn't quite work. We fit in this sort of biobanking aspect. It's great because we're now in the right animal. We're looking at the correct condition. The issue is we're only looking at one time point. That's when someone's passed away. And normally it's at the end of or when someone's had that particular condition. Also, we receive tissue that's not living, so we can't give an intervention. But I'll touch on how that's changing a little bit as well. Then you've got epidemiology. Epidemiology is great because we can have a look at big groups of people, we can find big trends, but it takes a little while to then actually test those hypotheses. We have to do these 3 steps to prove what we think we found in epidemiology. So that sort of all works, that's our arsenal of research. And what we're trying to do with all these things is start issuing clinical trials. We have a drug that we think is going to work. We're going to issue that drug in people with that relevant condition, and we're going to see what happens. The difficulty is, what we're looking at is the brain, and you can only do so many measurements of the brain in life because you need your brain. And recruitment becomes a little bit difficult because it's a new drug, so people might not be willing to use it, and it's incredibly expensive. So what we're trying to do is tweak these bits on the left-hand side so that we don't waste money on incorrect clinical trials. And so that's sort of where we fit in as biobanking. And what's quite good at the moment is we're getting new techniques where these drawbacks tend not to apply anymore. One, because we can now, when we receive a donation, we can actually grow some of these cells in a dish, so we can start to test a treatment. So that's great. And we're actually widening our eligibility so that it's not just people with Parkinson's, it's people without. And what we tend to find is someone may not clinically have Parkinson's, but they have the beginning of Parkinson's pathology in their brain. So we're getting different time points of the condition as well. So that's where biobanking is really sort of coming to the fore at the moment as an area of research that's going to sort of help us get to the right clinical trials quicker. And a little bit on our processes. So that's who we are, what we do. And I sort of want to speak a little bit about if someone signs up to the brain bank, what's involved with that process. So we recruit quite widely, and what we're different from other brain banks is we want anyone who feels that they can donate to donate. So you don't just have to have Parkinson's, you don't just have to have MS for our other recruitment scheme. We want anyone who feels that they can to donate from anywhere. You don't have to sort of be on a clinical trial or anything like that. We facilitate donations from the bottom of Cornwall to the tippy top of Scotland. We do Jersey, we do the Isle of Man, we do the Isle of Wight. And what we're trying to do with that is get a true understanding of the variability of the condition that is Parkinson's. And so we don't want to be restricted by saying you have to be eligible to be on a clinical trial. We do work with clinical trials. So often they'll have that clinical trial process and then they may also sign up to donate their brain. But we want to sort of encourage as many people as possible to donate. When someone signs up, we tend to leave them alone. We're not asking you to have any more hospital appointments. We're not asking you to do any extra scans or anything like that. That might be involved with the clinical trial. Essentially, you read a book, 16-page book. You say that you've understood what's in that book, and you fill out 3 forms, and you say, yes, I'm willing to pledge to donate my brain to research. And what we then do is every 2 years, we send out a questionnaire to say, what are you doing in daily life? How are your symptoms? And that sort of thing. Things that might not get popped onto the medical record. Personal experiences like that is what we're trying to gather, because we will get the medical record. So we're trying to have a 2-pronged approach there, so that sort of subjective and that objective approach. We bring those all together, we keep in contact with people through newsletters, but there's no sort of extra added sort of toil to signing up to donate. And then, when someone passes away, who's decided to pledge their brain, what we do is we ask that we're called, and then we look after everything else. We have an on-call 365 days a year on our emergency phone, and then we jump into action, we liaise with all the teams that we need to. Those teams being, this is normally where I do my long little winded bit because I like to show how much we need to do in 48 hours. But so essentially someone will need to pass away in the community. They'll need to have their deaths verified by a district nurse. That district nurse will pass that information onto the GP surgery. The funeral director will attend site. They'll take that individual into their care. That funeral director will take them into cold storage within 4 hours. The GP surgery has to have a doctor on standby who's sort of seen that patient in life. They have to propose the cause of death. That cause of death gets proposed and given to a medical examiner in an independent hospital. They will then say, does this make sense? If it does make sense, they'll call the family and say, are you happy with what's being proposed on that? That will then get agreed. At that point, we're legally allowed to proceed with the donation. We then have to make sure the local hospital has staff and capacity to assist us with the donation. Funeral director has to be available to transfer that individual to the local hospital. Medical courier is on standby to take the brain from wherever that hospital is in the country to us in London. By the time that happens, I clock off and say, see you later, technical team, and they all sit in the laboratory waiting for that donation to arrive at whatever hour it does, and they will then quickly prepare that ready for research. Has to happen within 48 hours. You can see there's quite a bit there, but we do facilitate about 70% of donations that people decide to pledge. That's not always because these sort of break down, as it were, the processes. Sometimes someone might not be eligible, but we do have a pretty high success rate, and we have a team on call at all times to be able to do that. And we always feed back to the family on how successful it's been or if it hasn't been. I'm not sure how I'm doing for time. Okay, we're good. We then preserve that tissue essentially by hemi-dissecting the brain, and then we do the first sort of gory bit of the presentation. I apologise. We then do sagittal slices of the brain. So what we're doing is essentially like an MRI, doing this way, and what we're trying to do there is be able to preserve one of those slices in formalin and one of those slices frozen. And so what we're trying to do there is have access to some of the molecular things. That's what we do with the frozen tissue, so we can look at the DNA, the RNA, and the protein. And the fixed stuff is how we do the actual diagnosis, because that's sort of the tried method. So we do that all within 48 hours. It looks like this. For the fresh tissue, what we do is we lay this on a grid. What we don't do is we try and look for the particular areas at that point in time, because we want to preserve the tissue as quickly as possible. And so we essentially put it on a grid system. This would be now slice P2, B3 there, and then we'll later go back and have a look at that area of the brain and say, B3 is perhaps the hippocampus or the amygdala. And then we'll have that in our library of brain tissue, essentially, in our 32-80 freezers. And when someone requests to look at a particular area of the brain, we can search our database, look for those particular areas, and pull it out for that researcher. So it's a nice big library, essentially. And then what happens with the fixed half is we bring all this information that we've collected in life through either medical records or through questionnaires. And what we're looking to do is prepare the tissue into thin slices. This is a nice pretty stain. It's a nice pink one. We call this hematoxylin and eosin. Essentially what this gives us is just the structure of the cells in the brain, and that's the one that the neuropathologist will always want to look at. And then we go hunting for particular proteins like alpha-synuclein, which is present in Parkinson's pathology, and that's how the neuropathologist will say, this is a brain with this type of pathology or that type of pathology. So when the researchers receive it, they know what they're doing the research on. Someone may have presented, which leads me nicely to my next slide. It's like I've prepared these in advance or something. Because what often happens is, at that neuropathology step, that's our clinical audit step. And so we do have some individuals, and they'll present this as a female, 76 years old. She had Parkinson's for 9 years. Her first presentation was sort of small handwriting and a gait impairment and an occasional tremor. She was diagnosed with tremor-dominant Parkinson's. When we received that donation, we had a look. And yes, in this staging of 1 to 6, we had this BRAC stage 6. And we said, yes, that was advanced Parkinson's. And there was also a small amount of Alzheimer's pathology. And so that's a different protein that causes that. But anyone over 60, we tend to start to get that Alzheimer's pathology. It doesn't mean you have the clinical symptoms, but there's that sort of pathology present in the brain. So that was someone who presented with Parkinson's. We did the clinical audit. They had Parkinson's. We then have other individuals, like this 73-year-old gentleman, who presented with memory disturbance and problems and sort of balance with walking, postural tremor on his left-hand side. And it was diagnosed with idiopathic Lewy body disease. So that was sort of Lewy body dementia because of that presentation with the dementia early on. But when we looked at the brain, what we found was actually high-stage Alzheimer's pathology and very moderate stage Parkinson's pathology. So this individual actually had mixed dementia. And so if we started to do work on this brain, assuming it was that Parkinson's diagnosis, we would start to get the wrong inferences from it. And so that's what's really important in this step is clinical symptoms. Doctors are fantastic, the neurologists, but they're not perfect. And so that's why we need this clinical audit step. We even have occasions where we have individuals be diagnosed with Parkinson's disease, like this 71-year-old female. She had a tremor in her left hand. We looked at the brain. There was no pathology at all. It was a very small amount of Alzheimer's pathology, but as I said, that was to be expected of someone who's over 60. And so why is that? And so once again, we want to know this so that we can now have a look at this individual's brain and say, okay, so what might have been causing that pathology that's given us the tremor but nothing else? But also, we don't want to give this to a researcher who thinks that they're working on someone who has Parkinson's, because once again, the wrong inferences will be made.
On an earlier slide, I know on these three, Alzheimer's disease was something that was found post-mortem and wasn't diagnosed pre-mortem. But on an earlier slide, you said that if someone has Alzheimer's disease diagnosed, their brain is not eligible for the bank.
So that's...
Why is that?
So yeah, of course, yeah. So we've recently changed our eligibility criteria. So because we've got some funding from a big international project where we're looking at people with co-pathologies as well. So now if someone has a diagnosis of Parkinson's with another sort of diagnosis, we would accept that. The only reason we wouldn't accept a purely Alzheimer's diagnosis is we only have one sort of pot of money. And our focus is on Parkinson's and related conditions, and there's other brain banks that focus on Alzheimer's. So if that was someone wishing to donate, we would say, try the Bristol Brain Bank or try Newcastle or something like that. So that's sort of and so often if someone's signed up throughout life and then they pass away and we find out they had Alzheimer's maybe at the point of death, we'll still facilitate that donation and then pass it on to one of the other brain banks to do the work.
So you do coordinate?
Yes, yeah.
Interesting that the two combined like that.
Yes, yeah. And so, I mean, they're sort of conditions that will obviously both be heavily linked with aging, and so that's why you'll find them often cohabitating together, but often one will sort of have their clinical symptoms more than the other. And so this is where the doctor's appointments and trusts having better sort of contact time with patients is more important, because what we're trying to understand is someone just said, oh, you have dementia or you have Alzheimer's. It's what we're trying to find is those intricacies between the two and then try and see how that matches up with the pathology. But to say 85% of the time, neurologists are right. And when they're wrong, they tend to be in the right ballpark. And so they'll often say, when someone say has Parkinson's or one of these Parkinson's plus conditions, they'll say it might be of academic interest, which diagnosis is, but it may not have too much effect on the clinical course or something like that. And that's sort of where we fit in, because what we are trying to do is understand how these are different, because we can't give someone with one condition the same treatment and expect it to work. And then, very importantly, we provide that tissue to research. So once we've got that, we call it a synoptic report, not a diagnostic report, because technically the diagnosis has happened. So we're doing a synopsis of what we find in the brain. Once we have that, we make sure that we get that tissue to research as quickly as possible. From the moment a researcher applies, we make sure they get that tissue within 4 weeks. And then from that point, we check in on them yearly to ask them, what research have you done? What can you feed back to us so that we can store it in our database? We support about 40 to 50 projects a year across the world. And so, how are we doing for time? Oh, we're running. You might have to come back a little bit later for me to finish the rest of the presentation, folks. There we go. So to sort of say where we fit into and why looking at the brain itself is important, because obviously it does feel like, well, why aren't we trying to make more inferences from people while they're living and that sort of thing? And obviously it is difficult because the brain is so important to us living, so we have to access it later on. But to show why looking at the brain remains so important and why the charities are so invested into sort of looking into it, you can sort of walk through the progress in understanding of Parkinson's. You know, we go to 1870 when there was that first definition of what it was, and then it took another 70 years before we started to figure out that this very small region, embedded in wax here, right here at the back of the head, we started to notice that in individuals with Parkinson's, there's normally a black line here in the brain, and it wasn't there. That's interesting. What's going on? Took another 20 years to find out, oh, there's these little, there goes a balloon. Oh, there's these little sort of black things inside the nice big neuron, and what's going on there? It then took us another 40 years before we figured out, oh, those things, what they are is they're dopaminergic neurons. And so they're neurons that basically like to have that, they like to send dopamine from here to send it to other neurons to make that message sort of happen. Often it's an inhibitory message to say basically don't move. That's where you get that tremor from. And then we got a few serendipitous sort of bits of research that came out in the 70s and 80s where essentially there was a chemist in a lab in America, and he liked to play with the drugs that he was making, and he took one drug and it turns out he got Parkinson's from using one of the drugs that he used because he made the drug incorrectly. And what we found out, what that was doing, it was inhibiting mitochondrial activity in areas of the brain. So we go, the mitochondrial activity is going down, and that sort of led to Parkinson's symptoms. Maybe there's something involved with the mitochondria there. In 1992, it was the first time we actually started to look at someone's clinical symptoms and match them to what we find out in the brain. And for a while, that was called the Parkinson's Disease Tissue Bank Criteria. And that came from work that was done via the Parkinson's UK Brain Bank, supporting clinicians to understand what was going on, what they were seeing in the clinic, and how that was referencing to the tissue. And only about 20 or almost 30 years ago, we discovered what was actually those little black clumps in here was actually something called alpha-synuclein, that's a protein. And so that's where we sort of got to just looking at the brain. We then started to want to say, okay, someone might have advanced Parkinson's or not advanced Parkinson's, how does that reflect itself in the tissue? And so we had a staging system come 2003 by looking at the brain. In 2013, by using tissue donated to the Parkinson's UK Brain Bank, we looked at that protein that was in those clumps, and we started to figure out what's so special about that protein in someone with Parkinson's, because it's a naturally occurring protein, it should be in your brain. Turns out it's misfolding. 2013, so about 13 years ago, we started to figure out there's about 4 different ways that this protein likes to fold up. Some are good, some are bad, some cause it to clump up and to sort of start to kill the neurons. And we started to try and figure out how they're different. And only about 10 years ago did we figure out how to detect that protein in someone who's actually living. And so that sort of shows you where we're progressing with trying to understand Parkinson's. And about 7 years ago, we started to figure out 96 different genes that might have a role in Parkinson's. Not in the sense that you have the gene and you get Parkinson's, but there's an association. And most of life, unfortunately, isn't as simple as you have this gene, you get blue eyes. It's an interplay between your genes, your genotype, and what you have in your environment. That's not just sort of whether you lived next to a field or you lived in the city. It's how stressed were you, it's how much sleep did you get and that sort of thing. And certain different genes mixed with certain different phenotypes are likely what then cause individuals to have Parkinson's. But understanding that interplay and understanding what these 96 genes actually do is an entirely different thing. Because we know there's 96 areas of the DNA that do something, but we don't know what those areas do. And so that's where we're trying to understand those now. Well, I think I'm going to have to, 4 minutes. So the most important bit for me after we receive that donation is reporting that back to the family. And we often will host family members who have had their loved one donate to the tissue bank. And we also let them speak with Professor Gentleman, our neuropathologist, to understand what's going on in the report. It's gobbledygook to me, so I imagine it's gobbledygook to most people. And so often our neuropathologist will speak through that with the family so they can actually understand what's going on in there, because we give them the entire scientific report. And then to sort of say what we are as a tissue bank in numbers, we've received 1,600 donations. And to show you, compared to the other brain banks in the UK, with this big blue one, this is all the donations that we've received from individuals with Parkinson's, and you can see a vast majority of those are done through us. The other brain banks will have received these donations normally because someone may have been diagnosed with something else, but it turns out they have Parkinson's pathology. We do have the lowest recovery times in the country, not quite the world, because there are other countries that have different systems, but so we pride ourselves in sort of being around for so long and receiving the tissue in the quickest way possible, which means it's of the most use to research. And I might call it there, and I'll go over some of our research outcomes in one of my later talks. How does that sound? Leave you hanging. But I'm very happy to have a chat and sort of speak about what we do. But the big thing, if you want to sort of see some of the work that we are supporting through these donations, there's the new study that's called the Landmark study. And essentially what we do, don't know how everyone knows their sort of genetic dogma, where we go from DNA to RNA to protein. But that's how we try and understand conditions. We try and understand, is there certain genes that cause someone to have this condition? If so, how are those instruction manuals being read? Because you're only reading certain parts in different parts of the body. And then from those bits that's being read, what's actually being turned into a functional protein. So the proteins are the doing parts in the cell. So that alpha-synuclein is a doing part of the cell, but it's sort of congregating and causing the cell to die. And so what we're trying to understand is the condition on all three of those levels. And normally we might give the tissue out. One researcher does one group, one researcher does another bit of research, but they're all doing it on slightly different platforms, which means you can't quite compare the results. And so there's a funding initiative now between Parkinson's UK and 3 or 4 pharmaceutical companies with this Landmark study, where they said, we have all these brains here to look at, and they're looking at all of these, and they are looking at that on the DNA, RNA, and protein level. So they're trying to get this complete sort of atlas of Parkinson's in that sense, to then find suitable drug targets, which is where the pharmaceutical companies are very interested, because they want to basically say, give us a set of targets and we'll fund it immediately. And so I touch a little bit on that later on the slide, but if you're interested, I would look up Landmark UK study, Parkinson's UK, and you can see some of the research we're supporting there. Thank you for your time, folks. Half an hour flies by. Don't know how much more I've got. A bit.